The objective of this practical is to determine the coagulation or clotting time in human body. Now we will understand the principle of this practical. Clotting time is the time taken by a specimen of blood to clot or coagulate. It ranges between 3 to 10 minutes. Whenever a blood vessel is ruptured, a clot is formed. So, the clotting time is said to be the time period between the appearance of blood and the evidence of formation of the first fibrin thread. Clotting takes place in three essential steps. First is formation of prothrombin activator. Second is formation of thrombin. Prothrombin activator catalyzes the formation of thrombin. Third is conversion of fibrinogen into fibrin thread. Thrombin acts as an enzyme to convert fibrinogen into fibrin monomers or threads. Now we will talk about blood clotting or blood coagulation. It is the third mechanism of hemostasis. Blood remains in its liquid form as long as it stays within its vessels. If the blood is drawn from the body, it thickens and forms a gel. Eventually, this gel separates from the liquid. This gel is called a clot. A clot contains a network of insoluble protein fibers called fibrin, in which the formed elements of blood are trapped. The process of gel formation is called clotting or coagulation. It is a series of chemical reactions that result in the formation of fibrin threads. There are two conditions. First is thrombosis. If blood clots too easily, the result can be thrombosis, which is a clotting in an undamaged blood vessel. But if the blood takes too long to clot, hemorrhage can occur. Clotting involves several substances which are known as clotting or coagulation factors. These factors include calcium ions, several inactive enzymes that are synthesized by hepatocytes, which are liver cells. They are then released into the bloodstream. It also includes various molecules that are associated with platelets or released by damaged tissues. Clotting is a complex cascade of enzymatic reactions in which each clotting factor activates many molecules of the next clotting factor in a fixed sequence. Finally, a large quantity of product, which is the insoluble protein fibrin, is formed. Clotting can be divided into three stages. Stage 1 involves two pathways which are called the extrinsic pathway and the intrinsic pathway. These pathways lead to the formation of prothrombinase. Once prothrombinase is formed, the steps involved in the next two stages of clotting are the same for both the intrinsic and extrinsic pathways, and together these two stages are referred to as the common pathway. In stage 2, prothrombinase converts prothrombin, which is a plasma protein formed by the liver, into the enzyme thrombin. In stage 3, thrombin converts soluble fibrinogen, which is another plasma protein formed by the liver into insoluble fibrin. This fibrin forms the threads of the clot. Now, what are the requirements for this practical? In order to perform this practical, you will need blood lancet, spirit or alcohol swabs, stopwatch, and a capillary tube. What is the procedure of this practical? First, get a deep finger pricked and allow the blood to flow freely without squeezing. It is important to prevent admixture of tissue fluid with the blood. Then discard the first 2-3 drops of blood and allow a large drop of blood to form. Then fill a capillary tube with the blood by dipping one of its open ends in the blood drop. The blood rises and fills the capillary tube by capillary action. Then break off 1 centimeter of the capillary tube from one end after every 30 seconds and look for the formation of fibrin threads between the broken ends. Note the time as soon as you see the fibrin threads. This is the clotting time. Now, what is the clinical significance of this practical? The clotting time is prolonged because thrombin cannot be normally generated, although the bleeding time is normal. The bleeding time here reflects vasoconstriction and platelet plug formation independently of clot formation. Clotting time is increased in conditions such as hemophilia, which occurs due to deficiency of clotting factor 8, Christmas disease, which occurs due to deficiency of clotting factor 9, von Willebrand's disease, in which both the clotting factor and platelets are qualitatively deficient, severe liver disease, vitamin K deficiency, and anticoagulant therapy. Now, here is one question. What causes decreased clotting time?